Hello, and welcome to the protein video of our biochemistry module. And here we'll learn about what proteins are, how they're folded, and how they're created, and we'll learn about the peptide bond, my favorite. So proteins are really important when dealing with enzymes. We'll learn how the specificity of enzymes um, is necessary and how they're just made up of protein. They make our structure in our bodies based on the types of protein, especially like in our hair and nails. Um, and they definitely serve as receptors, which we'll learn especially down the road in allergies with like histamine receptors, and viral receptors and receptors in the cell to let nutrients through the cell membrane, um, which goes into transport. All right, and so they also play a major role in the extracellular matrix, okay? And that just means stuff outside of the cell. The cells kind of project branches of proteins out into that extracellular matrix, so outside of the cell, extra. All right, so with that, how are proteins made is what our next question would be. And their simplest building block, like a sugar, like a saccharide is a monosaccharide, their simplest building blocks is called an amino acid. And you've probably heard of aminos. And if you're a weightlifter, you know about getting your amino acids and protein. And an amino acid just means that it shares this sequence. And if you look, they all share that same um, structure. And it's only the structure in blue that's different, and that allows them to differentiate. Um, and remember, you can get polarity on this nitrogen, and other times you can get polarity here. You can get a negative polarity on this side. And so this one just goes to show you how important those endings are in the whole scheme of what this protein can do and what is it made of. Okay, so though that special nitrogen bond is what makes that nitrogen there is what makes it a amino acid, and that nitrogen there bonds to the hydroxyl group of the other side, and it creates a peptide bond. A peptide bond. You want to remember that ideally for the rest of your life. I missed out on an opportunity in one of my job interviews about what a peptide bond was, and right now I'll never forget it again. Okay, so this is actually what ribosomes make. We'll get to that in chapter 3. So ribosomes are a little tiny factory inside of the cell, and they create protein, and they create protein via a peptide bond. Okay, and if you uh, don't have proper shaping of your protein and you have an error, either in the genetic code or somewhere down the line, you can get improper folding, you can get an improper protein, and that's what happens here in what's known as sickle cell disease. And these characteristic cells are in a sickle shape, and that sickle shape is due to an improperly folded protein. What do I mean by improperly folded protein? Um, well, it either got a bad uh, sequence and the peptide bond wasn't correct, and so it stopped abruptly, or we have a mistake in one of these levels of protein structure. And so we have our primary, or a 1 with a circle. We can have our secondary, or 2 with a circle, 3 circle, or 4 circle. Okay, and so that's primary, secondary, tertiary, and quaternary. And our primary structure is just how the amino acids are linked together. And so if we have a problem there, we have a problem with our primary structure. If we have a problem with how it is folding due to its hydrophobicity or hydrophilicness, it will not create beta pleated sheets properly or alpha helices. And if you finally can't fold those properly, you're probably not going to get a good tertiary structure, which is mainly most proteins. And then sometimes you can get a quaternary structure if it consists of more than one amino acid chain, which isn't common, but proteins like hemoglobin uh, is a, has a quaternary structure. All right, and so all these proteins are folded in such a way that they allow only what they need to into what they need. So if it's a receptor, it only allows what they receive into their active site. Okay, and what do I mean by that? Um, as an example, there's things called channel proteins, and these channel proteins um, let things in and out of a cell. And what's a cell? Well, we'll get to that in just the next lecture. And so what goes in and out of here, though, is very selective. Okay, and so not everything can get through, and that's what protein job is for. All right, and so protein structure leaving here today is very important. They are used in enzymes. They are formed by a peptide bond, and they are made by amino acids. Those are the things you want to take home with protein. All right, and that concludes our biochemistry module, and we'll see you in the next one.